Hi, and welcome to my presentation on microbial ecosystems. So before we get started talking about microbes specifically, we'll just talk about some general basics of ecology, starting with some terms. So our first term is a population. A population is a group of organisms that all belong to the same species that live together in the same place at the same time. So for instance, in El Paso, you have a number of different populations. You have all the humans in El Paso, that's one population. You have all the domestic cats, that's another population. The domestic dogs form a population. The pigeons form a population. So however many species you have living here, that's however many populations you would have. One for each species. Then all those species in different populations are also living together in a community. So in ecology, a community is multiple populations of all different species that live together in the same place at the same time. So all of the organisms that live in El Paso would form a community together. Then above the community level, you have the ecosystem. An ecosystem is both the communities that live in an area and the physical environment. So if you're adding in the buildings and the ground and the mountains, that's the ecosystem of El Paso. Well, all of those physical things and the communities would be the ecosystem for El Paso. Although in general, ecosystems are pretty large, so El Paso would not be large enough to count as a whole ecosystem. In an ecosystem, you always have the organisms interacting with their physical environment. So the environment, of course, impacts the organisms that live in that environment, but also the organisms will impact the environment, for instance, by causing erosion. And in an ecosystem, you'll have not the exact same physical conditions everywhere in the environment, and so that means you have habitats there as well. When one part of an ecosystem has conditions that are a bit different from another part of an ecosystem, those are two different habitats. Each organism in the ecosystem is going to be better adapted to some habitats and less well adapted to other habitats. So for instance, we are well adapted to living in the habitat of our buildings. We are not well adapted to living in the habitat of up on the mountain. Although we technically could do that if we wanted to. So a lot of the time you'll have different species living in different habitats all within the same ecosystem, which means that you can have different communities in different habitats, or every habitat would have kind of its characteristic community that would be different from a nearby habitat. In this image here, you have kind of a summary of those terms. So the lowest level of kind of organization in ecology is the individual, just one single organism. Then if you get a group of individuals, all of the same species, living together in the same place at the same time, that gives you a population. If you add in populations of different species, that gives you a community. Then when you add in their physical environment, that gives you the ecosystem. And in this particular diagram, we have kind of a river running through a grassy area, but there's also some more deserty areas right by the river. So that would be at least 3 different habitats. You have the river bank, the river itself, and the grassy area beyond the river. Then if you wanted to keep going to higher levels, you have a biome, which would be like a particular type of ecosystem, and the communities, well, and of course the organisms that live there, that is kind of repeated in different places across the world. So for instance, a desert biome, we have one here, and in other places in the world where they have deserts, they also have the desert biome. Or you could also have like a tropical rainforest biome, or a temperate forest biome, or a tundra biome. And then the biggest level of organization in ecology is the biosphere, which is just all of the organisms on Earth and the places where they live. So kind of the part of Earth that supports life. And we talk a lot about biodiversity in ecology. There's a couple of different ways that we can measure biodiversity. The first is to look at the species richness. which is just how many different species you have living in a particular community. You could also look at the species abundance, which is how many individuals you have for each species in that community compared to how many individuals you have for the other species in the community. So, like, do you have just one species that's super common and the other species that are there are kind of rare, or is it more like an equal mix where every species has a similar representation as the others? For microbes, it's hard to measure these things a lot of the time. For species richness, we can try to measure that using our RNA. If we just take a sample from the environment and sequence the rRNA in that sample, we can get an idea of how many different species are there. And actually, if we're really careful when we do that, we can also get some idea about the species abundance as well. We might be able to notice that one particular sequence of rRNA is real common, another is not very common, and that can kind of tell us that, the first species that has the real common rRNA sequence is more abundant in that environment than the other species that has the less common rRNA sequence. In general, we like to have a high species richness and kind of an even species abundance to have a very diverse community or a diverse ecosystem. And a lot of the time, those things are kind of inversely related to each other, where if you have a huge abundance of a particular species, that is frequently associated with not having very much species richness in that particular environment. So usually if you have a lot of richness, then you'll have kind of more balance in terms of how many individuals for each species are there in that environment. If one species or a couple of species are coming to dominate, that usually means that the environment is just not very well suited to a lot of different species. Only those couple can really thrive there. So that means that there's just probably not that many other species that can survive there at all. That's not necessarily the case, it's just common to find that trade-off. In any case, the species richness and the species abundance will always be determined by the conditions of the environment or the habitat where you're measuring. So that depends on the physical conditions of the environment and also, of course, what nutrients are available there and how much of those nutrients are available. So in this diagram, it's kind of illustrating the species abundance concept. In the first, example of a community, you have 4 different species of trees living here, and they're all kind of at an equal abundance. So you have about 4 individuals in this plot of land from each species. So the abundance will be equal. In the bottom diagram, it's a different situation. You technically still have the same species richness since you have the same 4 species of trees living there, but most of these trees would be this type of tree, which is labeled tree A up here, or species A. You have a lot of individuals belonging to that species and just a few from the other species. So here the species abundance is heavily skewed to favor this particular species of tree at the expense of the other species of trees. So we would say in general that this is a less diverse kind of forest than the top forest, which is more diverse. And on the bottom, you have kind of an illustration of how species abundance and species richness can change really quickly in an environment, especially for microbes, because their generation time is so short. So this is kind of a body of water where this guy is taking samples and then looking at the samples. Under normal conditions, the sample kind of looks like this under the microscope. You can't tell exactly what species are there, but you can tell there's a lot of different ones because you see a lot of different shapes. So there's a lot of species richness here and kind of a more even species abundance. So this is a pretty diverse habitat right now, or under normal conditions. But if you had maybe some polluted runoff into that water that brought kind of a nutrient influx, like maybe fertilizer ran off into that water, then you're going to have a lot more nutrients there. The autotrophs are going to start using those extra nutrients. They're going to have a population explosion. that's going to produce a lot more kind of fixed carbon for the chemoorganotrophs to use. So then they're going to have a population explosion, and you end up having a lot of species that kind of get crowded out and are not very abundant anymore. And then if you go and take the sample, it might look something like this, where if you're just sampling from the top of the water, you're having just a lot of autotrophs. So these are all cyanobacteria that have undergone a bloom or population explosion in response to an influx of nutrients into the water that would not normally be there. And they've really crowded out all the other microbes that you would normally find in that water. If you went deeper into the water, then you would find that it's a bunch of chemoorganotrophs, and if you went even deeper, you would find that the oxygen has all been consumed and you have a lot of anaerobes. In microbiology, we also group microbes together into guilds, not just into kind of populations and communities. So a guild is a group of populations of microbes that are, of different species, but they all have kind of similar metabolisms. They have kind of a similar strategy for getting energy out of their environment, and they would be using either the same nutrients or quite similar nutrients. So for example, you have a lot of different species of bacteria that are either sulfur reducing or sulfate reducing. They all use a pretty similar metabolism where they're using either sulfur or sulfate as an electron acceptor for anaerobic respiration and producing hydrogen sulfide by reducing either the sulfur or the sulfate. So their metabolism is quite similar, their waste product is the same, the nutrients that they use are not exactly the same, but they are similar. So we would group those different species together in a guild if they were living together in the same place at the same time. And it's very common to find microbial guilds in the environment. Each different habitat is going to have its own kind of set of conditions and set of nutrients that are available. So it makes sense that microbes of different species that can use the same nutrients are all going to kind of end up living together there in the same habitat if those nutrients are available there, and forming a guild together. In a community of microbes, you would usually have multiple guilds. It does depend on the habitat exactly and how many different nutrients are available there and what the conditions are, but usually you're going to have a number of different guilds in every community. And in the diagram is just kind of illustrating that. So for microbes, you would go from individual to a population, group of individuals of the same species living together. Then above the population level, you have the guild, which would be different species that live together that all have a similar metabolism. So you have a couple different guilds here, and then in a community, you would have multiple guilds. And all those microbes that are living together in that same place at that same time would be part of the community, and they would be arranged into different guilds. And sometimes you can have one microbe that participates in multiple guilds. So for instance, if you have a phototroph like cyanobacteria, that's going to be in the phototroph guild, but cyanobacteria also fix nitrogen, so that would be in the nitrogen fixing guild as well. And then in the ecosystem, you just add in their physical environment as well. Different ecosystems will also have different energy that's available in those ecosystems. Different habitats will have different energy sources that are available. You have kind of three basic types of energy sources that an ecosystem or a habitat could have. First, of course, you have sunlight, which is kind of the ultimate source of energy for most communities of organisms or most ecosystems. So for sunlight, the energy there is contained in the photons, which are the particles that kind of make up the sunlight. And phototrophic microbes, or of course land plants, would be able to use that energy in the photons to manufacture organic compounds. Next, you have organic carbon, which would basically just be organic compounds because organic compounds are always based on carbon. So with organic carbon, the energy is always going to be found in the chemical bonds that are holding that compound together. And chemoorganotrophic microbes would be able to use that carbon or those carbon-containing organic molecules as a source of energy to make, their own macromolecules for the cell and all the energy that they need. Organic compounds not only are a good source of carbon, they're also a good source of other nutrients like nitrogen, oxygen, sulfur, and phosphorus that all microbes will need to build nucleotides and amino acids. Of course, phototrophs can also, a lot of them can also use organic carbon in respiration to make energy and macromolecules. Usually, organic carbon is going to be coming from phototrophs. So usually you start out with an ecosystem that does not have organic carbon. It has inorganic carbon in the form of carbon dioxide, and then it has sunlight. And phototrophs will start using that sunlight as the ultimate energy input to make organic carbon, or to make organic compounds that contain carbon. And then chemoorganotrophs will take that organic carbon and use it as their own energy source. So ultimately, these chemoorganotrophs are going to be getting their energy from phototrophs, who are ultimately getting it from sunlight. So that's the case for most ecosystems, but not for all ecosystems, because of course some ecosystems don't have sunlight. So in ecosystems where you do not have sunlight, it's going to be chemolithotrophs instead that are ultimately producing energy for the entire ecosystem. A lot of those chemolithotrophs are also autotrophs, which means that they would be producing organic carbon themselves that would then be used by chemoorganotrophs. For chemolithotrophs, their energy source will be reduced inorganic substances. So that would be stuff like hydrogen sulfide, or ammonia, or nitrite, or iron, the 2+ ion for iron, which is ferrous iron. So those are all inorganic compounds that have, they're reduced, which means they have a lot of electrons. The energy in those compounds is going to be contained in the electrons. And then chemolithotrophs can strip those electrons off and use them to make ATP. And most chemolithotrophs will also use those electrons to make organic carbon or organic compounds that contain carbon. And then they're going to end up supporting chemoorganotrophs that are in the same ecosystem by providing them with organic carbon. So naturally, the ultimate sources of energy in an ecosystem would either be sunlight or would be reduced inorganic substances, which would be used by phototrophs or by chemolithotrophs to make organic carbon, which would then be used as a source of energy for chemoorganotrophs. In the diagram, it's kind of showing an example of an ecosystem and different communities and different habitats, different guilds that are there. So here you have like a body of water and the sediments beneath it. So this is all one ecosystem, but there's multiple habitats here. The sediments represent one habitat. Then in the water, you actually have a couple of habitats. You have what's called the photic zone, which would be kind of the area of the water where light is able to penetrate. So that makes a habitat that's nice for phototrophs. And then you have the oxic zone, which would be the area of the water where oxygen is able to penetrate, which makes it nice for aerobes. Sometimes you might have an anoxic zone as well at the bottom of the body of water where oxygen does not penetrate, and then that would be a nice place for anaerobes to live. In this case, the body of water is not that deep, so you do have oxygen penetrating all the way to the bottom. And then in the sediments, those are anoxic, so they're nice for anaerobes, not for aerobes. Light is entering this ecosystem and is the ultimate source of energy for this ecosystem. So in the photic zone, where you have the light, you're going to have phototrophs using the light, stripping the energy off of those photons and using it to make energy and to make organic compounds, which would be what this C6H12O6 is. This is, the chemical formula for glucose, a basic sugar. Then that's going to be feeding the aerobes in the oxic zone with energy. They're going to be using that glucose produced by the phototrophs as a source of organic carbon for themselves to build their own macromolecules. As all these microbes are kind of metabolizing, living and dying, as they die, their bodies will kind of fall to the bottom of the body of water, and that provides organic compounds to the anaerobes living in the sediments. So some of these guys would be using those organic compounds, but also a lot of these guys are going to be chemolithotrophs that are using minerals present in the rocks of the sediments as a source of energy. There's just, you usually kind of don't get as much energy that way as what you can get from sunlight. In addition to thinking about how energy travels in an ecosystem, we also think about how different elements travel through an ecosystem, both in terms of like the physical places that they move to, or how they move through different physical places in the ecosystem, and the different chemical formats that they take as they move. So basically which different compounds they're a part of. So if you're looking at nitrogen, that could be in the form of molecular nitrogen, N2, it could be ammonia, it could be nitrite, nitrate, nitrous oxide. There's a lot of different forms that it can take, even amine groups within amino acids. So each of those different chemicals and how nitrogen is moving between those different chemicals is also part of cycling through the environment for nitrogen. For any element that is cycling through the environment, both physically and through different chemical forms, you're going to have microbes playing a large role in the process. Maybe not for any element actually, but for a lot of elements. Specifically, microbes would be helping elements move through different chemical forms because they catalyze a lot of different reactions. So just taking the element in one compound and converting that compound to a different compound. And that whole process of elements moving through the environment physically and chemically is biogeochemical cycling. A lot of the time you have microbes playing a large role in cycling an element between different chemical forms, but you can also have the physical, I guess, characteristics of the environment playing a role there, or I should say the chemical characteristics of the environment playing a role, where you can have like lightning or high heat or oxygen. Those things can cause different chemical reactions to occur, and therefore they can drive biogeochemical cycling for different elements. A specific subtype of biogeochemical cycling that we think about a lot in microbiology and in ecology in general is nutrient cycling. Nutrient cycling is biogeochemical cycling for elements that are important to organisms because they need them as nutrients. So that would be stuff like carbon, nitrogen, phosphorus, oxygen, sulfur, anything that organisms require. So nutrient cycling is how those different nutrients move through the environment, both physically and in terms of the different chemical forms they take as they move through the environment. And again, you're usually going to have microbes playing a large role there in transforming elements between different chemical formats. We actually have a chapter about nutrient cycling coming up in the semester. This diagram is just kind of showing a basic view of the nitrogen cycle. So you kind of can start at the top with nitrogen in the atmosphere in the form of molecular nitrogen, or N2. Some of that nitrogen is going to end up diffusing into the soil. So then you have nitrogen cycling into the soil in the form of N2. And there you're going to have your nitrogen fixing bacteria taking it up and transforming it into ammonia. Some of that ammonia is going to be taken up by plants and converted into amine groups and amino acids. Some of that ammonia would remain in the soil, and you can have other microbes taking it up and using it. So some of them will take up the ammonia, specifically nitrifying bacteria, or the nitrifiers would take up ammonia and convert it first to nitrite, then convert it to nitrate. Then you have some plants that can use nitrate, so they might take up the nitrate and convert it into amine groups. You're also going to have animals eating those plants and taking the nitrogen into themselves and using it, for their own amine groups for amino acids. Then as plants and animals die, you're going to have different fungi and bacteria in the soil decomposing, their macromolecules, taking that nitrogen out of the amine groups and converting it frequently back to ammonia, where it would be converted then to nitrite, then to nitrate by different bacteria. Then some of that nitrate also is going to be used by denitrifying bacteria. They're going to convert it to molecular nitrogen, or to nitrous oxide, or to nitric oxide, but a lot of it to molecular nitrogen. In any case, all of those gases would enter the atmosphere, since they're gases, and then the molecular nitrogen, in the atmosphere is kind of completing the cycle there. This is not a complete picture of the nitrogen cycle. Of course, you have nitrous and nitric oxide that have entered the atmosphere at this point that we didn't talk about what will happen to them. Stuff will happen to them. They'll continue on in the broader nitrogen cycle. So that's just kind of a basic picture. So we talked about how different habitats within an ecosystem will be better suited to different communities of organisms, or to different species of organisms. For microbes, since they're so small, you actually have to get even finer grained than that. Microbes will not be living necessarily in specific habitats, but in specific microenvironments. A microenvironment is like a tiny, tiny little part of a habitat where you have conditions that are different from another part of that habitat. So for instance, a habitat might be kind of the open desert, but within that you're going to have different microenvironments. So if you are at a spot that's kind of far from any plants, that's one microenvironment. If you're right underneath the plant, that's a different microenvironment. You're going to have like dead leaves from the plant falling down there, stuff kind of accumulating there, and it makes for different conditions. And those conditions are going to lead to, I guess, some microbes being favored to live there and other microbes not being favored to live there. For every habitat, you're going to have really a lot of different microenvironments. Just any time that the conditions change, as you move from one part of the habitat to another, then you're looking at two different microenvironments there. And, including over an extremely small area. Actually, if you're looking at a single grain of sand, you can find different microenvironments even there in that grain of sand, which is what this graph is showing. This is like a sand grain, and what we're graphing here is the concentration of oxygen in different parts of the sand grain. So these numbers represent the percent of oxygen that's present in these different areas of the sand grain. In the atmosphere, oxygen is present at 21%, or 21% of the oxygen is, 21% of the atmosphere is oxygen. So on the outside of the sand particle, you have 21% oxygen. As you move kind of deeper into this sand grain, the amount of oxygen is going to decrease. So you have a zone where there's 15% oxygen, a zone where there's 10% oxygen, a zone where there's 5% oxygen, a zone where there's 1% oxygen, and in the very middle, a small zone with no oxygen at all, or an anoxic zone. So different microbes would be favored to live in different parts of this sand grain. In the very center, you could have some anaerobes living there. In these kind of areas where you have like just a little bit of oxygen, the 1 to 5% zone, you could have some microaerophiles living there that do need oxygen for their metabolism, but just can't tolerate, atmospheric concentrations of oxygen. And then in the outermost parts of the sand grain, you're going to have your aerobes living there. So even something as small as a grain of sand will have different microenvironments and different microbes living in them. Microbes are real tiny. So they're never going to be like affected by the habitat as a whole. They would be affected by the microenvironment that they're in. And they're always going to be kind of partitioning themselves into different microenvironments to find the conditions that are really right for them. Then a final thing to note about microenvironments is just that they can change very quickly. Things in a microenvironment can change very quickly. So for instance, you might think about if something happened to split this grain of sand in half, all of a sudden this anoxic zone in the middle, it's not anoxic anymore. It's got plenty of oxygen there. It's going to be, 21% oxygen if the split goes right through it. All your anaerobes are going to die, and your aerobes will move in and colonize the new area. If you're talking about a bush, if it rains, suddenly the conditions under that bush are very different than they used to be. You can frequently predict what microbes you would find living in a particular microenvironment if you know the realized niche and or the fundamental niche for that species of microbe. The realized niche is the conditions under which a species of microbe is able to grow at its optimal rate or as fast as it possibly can. So in the lab, when we grow microbes in the lab or when we culture them, we're usually trying to provide them with the conditions of their realized niche, just the perfect conditions for them that will let them grow as fast as possible. In nature, sometimes you have the conditions of the realized niche and sometimes you don't. Of course, the realized niche is going to be different for different species of microbes. But actually, if you're looking at a specific guild of microbes, usually the different species in that guild will all have about the same realized niche. Not necessarily exactly the same, but at least similar. Since they're all the guild members would have, a similar metabolism and need similar nutrients, that usually goes hand in hand with needing similar conditions to grow in optimally. Of course, since you don't always have the perfect realized niche conditions in the actual natural environment, sometimes you're going to have microbes growing in a place that's not quite optimal for them. And for that reason, we also have the concept of the fundamental niche, which is the whole range of conditions under which a particular microbial species can grow at all. So earlier in the semester, we talked about how different microbes would have different temperature requirements, and microbes will have an optimal temperature, and at that temperature they can grow the fastest that they can possibly grow. So that optimal temperature would correspond to their realized niche. That would be one of the conditions that makes up their realized niche. But then for each microbe, there was also kind of a whole range of temperatures that they could grow under, even though they can't grow as fast as at the optimal temperature. So that whole range of temperatures under which a microbe can grow at all would be part of the conditions of their fundamental niche. Of course, the realized niche and the fundamental niche don't just depend on temperature. They would also be depending on things like how much water there is, how much oxygen there is, the pH, the salt levels, the availability of different nutrients, the pressure sometimes. A lot of different conditions go into composing the realized and fundamental niche for different microbes. A lot of the time, you'll find microbes living in microenvironments that have the conditions of their realized niche. That's where they like to, or that's where they're able to grow the best, so that's where it's kind of the most likely to find them living. But you can also find microbes living outside of their realized niche in microenvironments that would correspond to their fundamental niche. You're never going to find a microbe living in conditions that are outside of its fundamental niche. You might find maybe some spores there in conditions that don't allow the microbe to grow, or kind of a dormant form of the microbe surviving there, but you're not going to find the microbe reproducing in any microenvironment that has conditions that are outside the fundamental niche for that microbe. This diagram here is kind of illustrating the concept of realized niche versus fundamental niche for barnacles. So these concepts don't just apply to microbes, they would actually apply to any species at all. So here we have like a rock coming out of the water, coming out of the ocean, and you have a low tide mark at the bottom of the rock and a high tide mark at the top. So as the tide changes, water would be moving up and down the rock between the low and the high tide marks. And you have a couple different species of barnacles growing on this rock. On the bottom, the big blue ones are Balanus barnacles, and on the top, the small brown ones are Cathylamus barnacles. And then in the pictures, it's showing you like what those barnacles actually look like. Then these arrows are showing the fundamental and realized niches for those two different species of barnacles. So first, if we look at the fundamental niches in brown, you can see that the fundamental niche for the Cathylamus barnacle is stretching all the way from low tide to high tide. What that would tell you about those barnacles is that they need to have some water on them, but not be underwater all the time. So they can't grow above high tide where you don't have any water at all, and they can't grow below low tide where you always have water no matter what. They need to be submerged some of the time and not submerged, I guess that's some of the time as well. In blue, you have like a smaller arrow. That is the fundamental niche for the Balanus barnacle. So it too cannot grow below low tide. It cannot be submerged all the time, but it actually can't grow up at the high tide mark like the Cathylamus barnacle can. That is outside of its fundamental niche. So it needs to have water on it for more time than the Cathylamus barnacle needs to have water on it. Or it has kind of a lower tolerance for different water conditions than the Cathylamus barnacle has. Then over on the right, you have arrows for their realized niches. So for the Cathylamus barnacle, the realized niche is mostly up at the high tide area and stretching down a little bit below that, which means that they would really prefer to not be submerged in water that much. If they're growing close to the high tide mark, that's when they're growing at their optimal rate. They can tolerate being lower down, closer to the low tide mark, but they just can't grow as fast there. So they can live there, but not as well. They can't prosper as well. Then the realized niche for the Balanus barnacle is actually mostly corresponding to its fundamental niche in this location. So it would prefer to grow beginning at about the low tide mark and going up most of the way towards the high tide mark, but not all of the way. In all of that range, it has conditions that are actually optimal for it to grow. So if you went to a different environment that had a different range of conditions, you might draw these fundamental niche arrows differently for the Balanus barnacle. But here in this particular environment, you draw them the same as the realized niche arrows, indicating that this barnacle can only survive in that area, and that area is also the area where it grows the best. And if you notice where they're actually growing corresponds to the realized niche. It does not correspond as much to the fundamental niche. So the Balanus barnacle is going to extend as far up the rock as its realized niche does. The Cathylamus barnacle is going to extend as far down the rock as its realized niche does. For that barnacle, the fundamental niche would actually extend all the way down to low tide, but you don't find those barnacles growing down there. Actually, if you looked close, you might find a couple, but you don't find very many of them. The reason is that if the barnacle was to start growing, lower down there towards the low tide area, then it's going to be competing for resources with the Balanus barnacle. And closer to the low tide area is where the Balanus barnacle is enjoying the conditions of its realized niche. So it's really able to thrive there, whereas the Cathylamus barnacle would be, or the Cathymalus barnacle would be outside of its realized niche there, which means it can't grow as fast. And it's basically going to be outcompeted by the Balanus barnacle. So you see the same thing with microbes a lot. They can grow outside of their fundamental niche, but if there's somebody else there who, for whom the conditions there would be the realized niche, then it's hard to compete. It's hard to compete, with somebody else who's growing in their realized niche when that same microenvironment is just part of your fundamental niche, but not part of your realized niche. So you usually find microbes kind of more limited to their realized niche than just growing wherever their fundamental niche occurs. Even when microbes are growing in a microenvironment that basically corresponds to their realized niche, their growth is usually limited by nutrient availability. Most ecosystems just don't have that many nutrients available. some ecosystems and some microenvironments typically have a lot, but we'll go through at the end of this chapter the different common environments to find microbes in, and most of them just don't have a lot of nutrients commonly. Like if you're a microbe growing underground or in the depths of the ocean, there's usually not that much around. And the levels of nutrients in a given microenvironment can kind of vary a lot. So they might be quite low for most of the time, but there might be times when you have like a sudden surge in nutrients. For instance, if we're talking about the bottom of the ocean, anytime that an animal dies, especially like a large fish, higher up in the ocean, their corpse will sink to the bottom. And right where the corpse falls, that'll create a microenvironment where you have suddenly a lot of nutrients available. So that would be like a nutrient surge until that corpse is decomposed and the nutrient levels go back to kind of more like normal. So most microbes need to be prepared for kind of long periods of low nutrient availability, and they need to be ready to take advantage of nutrients whenever they do come around. In general, microbes will store nutrients whenever there are a lot of nutrients. They will kind of make excess of whatever kind of food molecule they are making, or convert their nutrients into a form that can be stored. So an example would be like some microbes will take up sugars from the environment as a nutrient and convert those sugars into polysaccharides for storage. Like starch and glycogen would be polysaccharides that are used to store sugars. And then they can just kind of fill the cell body up with as much of that polysaccharide as they can. Then when nutrients run out in their environment, they can kind of raid their stored nutrients to continue making energy. And you'll usually see that microbes are not growing at a constant rate in their microenvironment, because the rate is going to depend on the nutrient availability. So growth will usually be pretty low in any environment that doesn't have a lot of nutrients. But when you get a nutrient surge, there's going to be a surge of population growth as well. As nutrients become available, the microbes will begin dividing and also storing as many of the nutrients as they can to use later. So one thing that scientists have realized by studying microbes in their natural habitats is that most microbes do not grow as quickly in nature as they do in the lab. When you're culturing a microbe in the lab, you're providing its kind of perfect realized niche conditions and the perfect nutrient levels, or, just very abundant nutrients. So microbes in the lab will grow at an idealized rate. If you find that same species of microbe growing in the environment, it will probably not be growing at nearly the same rate. It'll be much slower, even if the conditions of that microenvironment do correspond to the realized niche for the microbe, just because it's highly unlikely that the nutrient levels would be so abundant that they can support a really high level of growth. And microbes in communities always have relationships with each other. The most basic kind of relationship for microbes to have is a competitive relationship, where they're all trying to use kind of the same resources. So they're all limiting each other's growth. Competition plays a really large role in microbial communities. And in addition to kind of limited nutrient availability, competition is another reason why microbial growth in the natural environment is generally slower than it is in the lab. But in addition to competition, you also have cooperation amongst microbes. There's several different types of cooperation that microbes can do. First, you have syntrophy, which we had mentioned before. where you have a couple of microbes, a couple of microbial species or more working together to use a particular resource. So there's some microbes that are using some reaction for their metabolism that isn't favored to occur unless there's another microbe that's also using their product. Because any time that you're consuming a product, the reaction is just more likely to proceed forward. Then you also have microbes that have complementary metabolisms. So in that case, you have one microbe that is producing a metabolite, and then another microbe is using that metabolite as a source of energy for their own metabolism. A good example would be the nitrifying bacteria, or archaea, which are divided into ammonia oxidizers and nitrite oxidizers. So the ammonia oxidizers are taking ammonia and oxidizing it to nitrite, and nitrite oxidizers are taking nitrite and oxidizing it to nitrate. So they're both kind of working together to nitrify ammonia to nitrate, but the nitrite oxidizers are using the metabolite of the ammonia oxidizers, which is nitrite. Then finally you have symbiosis, specifically mutualistic symbiosis. There's actually the three types of symbiosis: mutualism, commensalism, and parasitism. So of course in parasitism you have one organism benefiting from the relationship, but it's harming the other organism. So that's not exactly cooperation, even though it does technically count as a type of symbiosis. Then in commensalism, you have one organism benefiting and the other organism not being affected, so we don't quite class that as cooperation either. In mutualistic symbiosis, that's what we usually think of when we think of symbiosis. That's where both organisms in the relationship are benefiting. So that is a type of cooperation, and there's a lot of different examples of symbiosis in microbes and I guess different forms that it can take. Wherever surfaces are available in a microenvironment, microbes will generally be living on the surface preferentially. They like to live on a surface when they can, which would be a sessile lifestyle when they're attached to a surface. Planktonic lifestyle would be if the microbes are kind of suspended in water, or technically in a gas as well, just not attached to something. In the sessile lifestyle, microbes are attached to some object which is serving as a surface for them. Actually, it can be any object at all that could act as a surface, just something that they're attached to. The benefits of living on a surface would be that, in general, surfaces will provide some type of protection to the microbe, as opposed to just floating around. Also, if you have a liquid flowing across the surface, it's going to be constantly bringing in nutrients. If you were a microbe that was in the liquid, then you might use up all the nutrients that are right around that local area of the liquid that you're in, and then you would kind of be waiting for more nutrients to diffuse over to you from the areas that you hadn't depleted yet. If you're attached to a surface and, say, water is flowing across that surface bringing nutrients with it, then as you remain stationary and the nutrients just kind of flow past with the water, and you just take them up as they're flowing past. If you were in that water, then the nutrients would not be flowing past you for you to pick them up. They would be flowing with you, and so you would only be able to get whichever ones were actually like right next to you in the water. When you have microbes living on a surface, it's common for them to form kind of a thick layer on the surface. If it is just like a little bit thick, then we call that a biofilm. But if it's very thick, then we call that a microbial mat. A microbial mat is something that would be visible to the eye. It's usually like centimeters thick at least. They can get quite thick actually, and it's basically just composed of microbes and their glycocalyces if they have them, and their waste products. In a microbial mat, you would usually see multiple layers, sometimes a lot of different layers. in each layer there's going to be a particular microenvironment. So every layer is going to have different conditions in it. Since every layer has a different microenvironment, you would find different species living in each layer, and different guilds living in each layer as well. In general, a microbial mat is going to have at least one layer in it that is really favorable for phototrophs or for chemolithotrophs to live in. And those phototrophs or chemolithotrophs would be producing organic compounds that are going to be used by the rest of the microbes in the mat to support their metabolism. So you just have some pictures of different microbial mats here, and you can see the different layers, as well as a diagram showing different layers of a microbial mat, and the way that different layers are going to have kind of different species living in them. In this particular one in the diagram, the top layers are green because there are phototrophic bacteria living in those top layers that are supporting the rest of the mat. So if you have a layer of bacteria on a surface that is not thick enough to be a microbial mat, then we would call that a biofilm. A biofilm is consisting of the microbes themselves as well as the stuff around them, which is mostly their glycocalyces, the slime layer glycocalyx, but it can also be other stuff. You'll have kind of, I guess, dead corpses in there, and just molecules that are released by these microbes as they metabolize and as they die. Usually you would have multiple species living in a biofilm. There could be exceptions to that, but it's usually a good number of species. And you usually also see multiple layers and multiple microenvironments, similar to a microbial mat, just not as thick, not as organized. The benefits of living in a biofilm for microbes are that they're protected from the environment, especially they're protected from any toxins that might be in the environment. They're also protected from being washed away. One challenge for microbes living on a surface, if there's water around, is, remaining attached to that surface. Since the biofilm is really slimy and sticky, it's easy for them to remain attached to the surface if they're in the biofilm. Also, some nutrients can become trapped in the biofilm, making it easier for microbes to gather them. In the pictures on the bottom, you have water running in a stream running over a biofilm. So this red stuff is actually the biofilm on rocks formed by bacteria, and then you have water running over on top of that. So the biofilm would be helping these bacteria remain attached to the rocks even though there's water going past at a good speed. Then up here you have like a cross-section through a biofilm in red. That is actually not the biofilm. That is the surface that these microbes are growing on. The biofilm is in yellow. And then in this image you have kind of a head-on view of a biofilm. And this has been stained so that the bacteria in the biofilm are glowing fluorescently, and you actually don't see the slimy matrix of the biofilm at all. So these black spaces would be the stuff of the biofilm, basically. So you can see that there's cells suspended in it, but a lot of the biofilm is actually not cells. It would be the slime layer glycocalyces, or maybe kind of degraded cell parts. Biofilms have become pretty important in medicine and in different industries because they form on equipment sometimes and it makes a problem. So for medicine, you can have biofilms forming on medical equipment, and especially implants, like anything that would be left inside of a person's body, like a catheter, could have a biofilm forming on it, which would be quite dangerous for that patient potentially. Also, there are some microbes that can form biofilms on the surfaces in the body without any type of medical implant. So of course the most common example would be the bacteria in your mouth that every day form the biofilm called plaque on your teeth, and then you have to brush it away each night. That's kind of normal. It's not the best, of course. That's why you have to brush it away, but it's not the end of the world. But sometimes you can have biofilms forming more internally in your body, which could be quite dangerous. An example would be Pseudomonas aeruginosa, which is a species of pathogenic bacteria that is kind of infamous for forming biofilms inside the lungs of people who have cystic fibrosis. Cystic fibrosis is a disease that has a lot of different symptoms, actually, but one of the most, I guess, common or prominent ones is just that you produce really thick mucus. So normally you should be producing mucus in your lungs that you then move up and out of the respiratory tract using the cilia on the cells lining the respiratory tract. But in cystic fibrosis, the mucus is just so thick that it's hard to move it. It's hard to move it up and out, which means that bacteria can get trapped there. And if Pseudomonas aeruginosa gets trapped there, then it can prosper there and form a biofilm, which is very serious and can kill you. Once you get those bacteria living in a biofilm inside your body, it's very hard to get rid of them. If you try to use antibiotic drugs, they usually won't penetrate the biofilm. Also, the same thing goes for your immune system. The cells of your immune system have a hard time accessing bacteria living inside a biofilm to destroy them. It's just hard to get through the biofilm. So that means that once you have a biofilm, it's really hard to get rid of it if it's inside your body. On the picture here, it's actually images of Pseudomonas aeruginosa forming a biofilm, not in a person's body, thankfully, but in green. I guess the cells are stained green. So when the cells are first added to the surface, you can see there's not that many. Then after a day, since conditions are optimal here, they've grown a lot and they've formed a kind of a solid lawn. And then as time progresses, the layers of the bacteria just get thicker and thicker. And with this particular species, they will usually form these kind of mushroom-shaped domes on their biofilms. It's kind of characteristic of P. aeruginosa biofilms. If this was actually inside of a person, it probably would not form quite that fast because conditions are not going to be perfectly optimal for the bacteria like they are in the lab. But even so, it can form, pretty quickly. In industry, biofilms can make a problem basically all over the place, but a really common type of equipment to form biofilms would be any type of pipe that's carrying a liquid through it. Also, any equipment that's kept underwater. Those are prime places where you would find a biofilm forming. If you have a biofilm forming in a pipe, it can get kind of thick enough to clog the pipe or at least reduce the rate at which you're carrying that liquid through the pipe. And then, of course, you can have bacteria or their waste products kind of breaking off the surface of the biofilm and ending up in whatever fluid is traveling through the pipe, which might not be a good thing depending on what it is. And then finally, just whatever surface the biofilm is on, it's pretty common for the bacteria in the biofilm to end up corroding that surface. So it just degrades your equipment and you have to replace it more quickly. Because biofilms are so, I guess, persnickety, such a thorn in our side, we've developed agents that can prevent biofilms from forming. In many cases, most of those agents would block microbes from communicating with each other. In order to actually form a biofilm, microbes have to release chemicals that send signals to other microbes. So they have to change their behavior to get ready for living in the biofilm. So if you can block that communication, then you can prevent the biofilm from forming in the first place. However, once you have the biofilm already formed, you can't really get rid of it using any type of drug. You have to physically remove it in general to get rid of it. So going back to microbial mats, there's a couple common types of mats that we'll talk about. The first is cyanobacterial mats. So it's kind of common to find microbial mats where different species of cyanobacteria are the main producers for the mat. They would be kind of living in the top layers of the mat where you have light able to reach, doing photosynthesis, producing oxygen, and producing organic carbon. Then those compounds would be used throughout the rest of the mat by other microbes living in the mat. Cyanobacterial mats would only be found in extreme environments. That would be like commonly hot springs or like right next to a hot spring. That's because if a microbial mat like this were to try to form just kind of on the ground, animals are going to come by and disrupt it because you just have kind of constant grazing anywhere that's like not extreme. So it's possible for these cyanobacterial mats to form in extreme environments because you do have some species of cyanobacteria that are extremophiles that can tolerate really high heat or extreme pH. And in these types of environments where you have weird pH or really high heat, like by a hot spring, that's where you would find these mats. The chemistry of the mat is going to be different during the day compared to during the night, in large part because cyanobacteria would only be doing photosynthesis during the day. So during the day, you have your cyanobacteria in the upper levels of the mat producing oxygen through oxygenic photosynthesis. Then typically in the lower levels of these mats, you would have sulfate-reducing bacteria that are taking sulfate, which would be present at a lot of hot springs, and reducing it, using it as a final electron acceptor for anaerobic respiration, and reducing it to hydrogen sulfide. So you have oxygen being produced at the top and hydrogen sulfide being produced at the bottom of the mat during the day. Then in the middle layers of the mat, you have aerobes that are also sulfur oxidizers, which means that they're using sulfur compounds, specifically hydrogen sulfide, as electron donors for respiration. So that makes them chemolithotrophs. So they're using hydrogen sulfide as their electron donor, and then they're using, of course, oxygen as their electron acceptor because they're aerobes. So they're going to be consuming both the oxygen and the hydrogen sulfide. Which means that once you get to those middle layers of the mat, you're going to see the oxygen levels drop off because the oxygen is being consumed by these chemolithotrophs. You would also see that the hydrogen sulfide levels are low in the middle, and then as you go down, they get higher because this hydrogen sulfide is being produced at the bottom, and then it moves up through the mat. But once it gets to the layers where you have those sulfur oxidizers, they're consuming the hydrogen sulfide. And converting it back to sulfate. So the hydrogen sulfide concentration in the upper level of the mat would be really low. You actually have some of those sulfur oxidizing chemolithotrophs that are able to move through the layers of the mat, so they'll just kind of hunt out like the optimal concentrations of oxygen and hydrogen sulfide each day. Then during the nighttime, the situation is going to change. You don't have photosynthesis happening in the night, so oxygen is no longer being produced in the upper levels of the mat during the night. So you're going to have like low oxygen throughout the mat during the night. Hydrogen sulfide, on the other hand, that's going to rise in concentration throughout the whole mat. So at the bottom, your sulfate reducers are still carrying on with their metabolism, producing hydrogen sulfide. Normally that would be consumed in the middle by the sulfur oxidizers, but the sulfur oxidizers actually require oxygen for their metabolism as well. So since there's no oxygen at night, they're not able to run their metabolism. They have to just kind of persist off of stored energy throughout the night, and hydrogen sulfide levels are just going to go up throughout the whole mat. So in the pictures here, you can see layers in a typical cyanobacterial mat. At the bottom here is where you have your sulfate reducers, and at the top where it's green, it's where you have your cyanobacteria. In the middle is where you would have your sulfur oxidizers. And then this graph is showing concentrations of oxygen and hydrogen sulfide throughout the mat during the day. So the top of the mat would be kind of right here. So you have high oxygen in the top of the mat, actually even higher than the air because the cyanobacteria are getting busy and making a lot of oxygen. Hydrogen sulfide would be high at the bottom of the mat. Then in the middle, you see oxygen dropping off and hydrogen sulfide dropping off as well. That's because both of those resources are being consumed by the sulfur oxidizers. And on this graph, you can see the pH changes throughout the mat. So different microbes at different levels would typically have different pH requirements. And the final type of microbial mat we'll talk about is the chemolithotrophic sulfur mat, which is found at the bottom of the ocean in a lot of places. In the chemolithotrophic sulfur mat, the main producer that you have would be sulfur oxidizing bacteria. They are anaerobic. Since it's the bottom of the ocean, there's not any oxygen around. They're also autotrophs, producing organic carbon for the rest of the microbes in the mat. And a lot of them are filamentous, so in these pictures, these would be images of the top of a chemolithotrophic sulfur mat, and you can see these kind of hair-like things on the top of it. Those are actually the filaments of those autotrophic sulfur oxidizers in the mat. And here's like a zoomed-in version of the filaments. When you have these mats on the ocean floor, you're always going to find beneath them in the sediment of the ocean floor sulfur reducers. So these sulfur reducers in the sediments would be using sulfur as an electron acceptor. They would be getting it from the minerals in the sediments, and they're going to be converting it into hydrogen sulfide. So you're going to have a lot of hydrogen sulfide at the very bottom of the mat. And as you go higher in the mat, the levels of hydrogen sulfide would go down because that hydrogen sulfide is being consumed by the sulfur oxidizers in the mat. They would be converting it either to a sulfur compound, to a sulfur compound, possibly sulfate. So these sulfur oxidizers would be using hydrogen sulfide as their electron donor for respiration. For their electron acceptor, they will usually use nitrate, and they're actually going to be collecting nitrate from the water at the top of the mat. So a lot of them will move up to the top of the mat when they need more nitrate. Then they have these vacuoles inside of them that they're going to fill up with the nitrate that they get from the top of the mat from the water. Once their vacuoles are filled up with nitrate, they will kind of dive deeper into the mat. They're going down to lower levels where you have more hydrogen sulfide. So once they're down at those lower levels, they're going to be using the hydrogen sulfide as their electron donor, using nitrate as their electron acceptor, producing organic carbon from carbon dioxide. Then that organic carbon is going to go to support the rest of the microbes in the mat. And once the sulfur oxidizers run out of nitrate, then they head back to the top of the mat to refill their vacuoles. So those are kind of some general aspects of microbes in their environments. And now we'll go through the most common microbial environments that we find on Earth. So the first common environment is just the soil. Soil all over the planet has microbes in it. Like literally no matter where you are, if there is soil, there are microbes living inside of it. Most soils have limited nutrients. Not every soil has limited nutrients, but most of them do. Specifically limited phosphorus and limited nitrogen. So phosphorus and nitrogen are kind of the most common components that you would add to the soil in a fertilizer. A lot of soils will have limited water, especially in kind of dry areas, of course. Soil usually has a good amount of oxygen in it, though. The exception would be if you have a soil that actually has a lot of water in it, that's like kind of waterlogged, then it's going to have less oxygen, just because oxygen is not very soluble in water. Most of the microbes that you would find living in the soil, no matter what type of soil it is, would be living kind of at root level. So in the zone where you have plant roots, that's the zone where you would find most microbes. There's also some microbes beneath there, just not as many. If we're talking about an arid soil, which would be a dry soil without a lot of water, you're going to have a lot of cyanobacteria living in it actually frequently. You also have other phototrophic organisms like green algae. Then you have fungi, lichens, mosses, and of course your heterotrophs that would be supported by all of those cyanobacteria and the green algae, who are your autotrophs, and the mosses, but that's a plant. In a lot of areas, you'll have structures called biological soil crusts, or BSCs. So if you've ever gone out walking and the top of the soil is kind of crunchy, that's a biological soil crust. So just a crust formed on the surface of the soil, but it's formed by microbial organisms actually. So generally by different fungi and by filamentous bacteria. Anything that has a filamentous shape to it can help form a biological soil crust. Those crusts are really important because they stabilize the soil. They make it so that the wind cannot blow the soil away. So if you lose the biological soil crust, then the area would become more vulnerable to desertification, which is when an area that is not a desert turns into a desert. So what would happen there is if a lot of the BSC is disrupted, then the soil is not stable anymore. When the wind blows, the soil is going to blow away in the wind, and it's going to blow to some new area. So you're going to have some other area that's kind of getting a lot of dust blowing in the wind, which can create like a dust bowl effect and make a desert where you didn't used to have a desert. So in this illustration here, you have kind of a typical soil environment. You're going to have grains of sand of different sizes. So if it's actually proper sand, then it's kind of big. If you have a smaller grain of sand, then we don't call it sand anymore. We call it silts. If it's even smaller, then maybe we call it a clay particle. And you'll have little pockets of water in the sand or in the soil, little pockets of air in there. And a lot of organic matter, just kind of little bits of organic matter throughout the whole thing, and then little colonies of microbes that are called microcolonies because it's not enough to actually see it with the eyeball, so it's not a proper colony, but you know it is a little cluster of microbial cells. Then on the bottom here, you have the biological soil crust. So this is a biological soil crust. They don't all look like this, just some of them do. And then if you were to zoom in on that with the electron microscope, you would see something like this. Particularly in this view, you can see the little particles of sand, and they're being bound together by these filaments, which are either from fungi or from filamentous bacteria in general. The next common environment is the terrestrial subsurface, which means the ground beneath the soil. Yeah, basically the ground beneath the soil. Terrestrial because it's kind of not underneath a large body of water. I guess specifically not underneath an ocean. So in the diagram here showing the subsurface, this real thin dark brown layer at the top, that is the soil. So everything beneath there is your terrestrial subsurface. It's a lot. It gets, very deep. There's no oxygen in there. It's definitely kind of an extreme environment because, there's no oxygen at all and there's very, very little nutrients. And the further deep that you go, the hotter it gets and the more pressure that there is as well. So if you're a microbe living in kind of the bottommost layers, then you need to be prepared to deal with a high temperature and possibly a high pressure. You wouldn't really find microbes living kind of everywhere throughout the subsurface, only in places where there's water. So wherever you have groundwater, you're going to have microbes. If there's literally no water at all, there's going to be literally no microbes. You also don't have any eukaryotes that live in the subsurface, only prokaryotes. Specifically, the types that we found are chemolithotrophs that are also autotrophs, and nitrogen fixers. Since there's very little nutrients around, the growth of microbes in the subsurface is going to be really slow. Even if the temperature is higher, which would generally speed up growth, there's just so little nutrients that no one can grow fast there. It takes a long time to get enough nutrients to build enough structures to actually like double in size so that you can divide. And finally, just we don't know a lot about microbes living in the subsurface. It's not the easiest place to investigate. And yeah, so we just haven't done a lot of those studies. But in this diagram, you can see kind of wherever there's water, little seams of water, or areas where the soil, not soil, where the rock is kind of, I guess, suffused with water. There you're going to find the microbes. Wherever there's water, there's microbes. The next common environment is freshwater, which would be, lakes, rivers, streams, ponds, stuff like that. Almost any body of water that's not the ocean, although you do have some lakes that are very salty, so if the water is salty, then it doesn't count as freshwater. The conditions in freshwater environments can be very different from body of water to body of water. It depends on a lot of different things, but one of the things that the freshwater conditions depend on is just how many pollutants are in that water. A lot of fresh water is contaminated by our pollution. And you have a lot of different types of pollution as well, so the effect that has on the water, it depends on what type of pollution it is. In particular, some types of pollution, like fertilizer, for instance, or just runoff from livestock operations, that contains a lot of organic material, which has kind of predictable effects on freshwater quality, and in general make it kind of hard for fish to live there. The temperature of freshwater depends on how deep the water is and, of course, what time of year it is. So usually you would have higher temperatures at the surface of the water, and as you go deeper, the temperature would drop. And during the kind of summer months, you get stratification in freshwater. If it's like standing water, so that at the top you'll have kind of one layer of water that's at a particular temperature. Then you'll have kind of multiple layers as you go down that are each have like a lower temperature than the one above. And you don't have much, I guess, traveling of the water between different layers. Most of the water would just stay in whatever layer it happens to be in. So the same thing would be for nutrients. Nutrients aren't going to, and oxygen aren't going to move between layers in the summer. Then in the winter, or I guess I should say in the fall, usually bodies of water that have been stratified in the summer will turn over, and you'll have water from the bottom kind of rising up to the top, and then it kind of displaces the water at the top, so the water that had been at the top will end up falling to the bottom, and you get kind of a circular turnover of the water, which means that water that was at different temperatures is mixing now. So you're going to have all the same temperature throughout the body of water, and you're also going to have nutrients and oxygen mixing as well. So parts of the water that used to not have a lot of oxygen or used to not have a lot of nutrients, now they're going to have more. So that's shown here with lake turnover. So in the summer in this lake, you'll have different layers depending on the temperature. And then in the fall, the layers will mix. At the same time, you're going to have the nutrients and oxygen in the water mixing as well. In general, if the water is moving, so like a river or a stream, you're going to have oxygen penetrating deeper into the water, which is again because of mixing. Anytime water is moving, you're going to have the surface bits kind of mixing more with the bits beneath and carrying oxygen down deeper into the body of water. The faster the water is moving, the further down oxygen is going to penetrate. You have a lot of phototrophs living in freshwater, specifically oxygenic phototrophs. Primarily that would be, of course, cyanobacteria and algae. You also have some anoxygenic phototrophs, you just don't have as many of them. So those phototrophs are going to be basically supporting the ecosystem in freshwater in general. So they're going to be producing organic carbon that is then going to be used by chemoorganotrophs that are living in that water as well. So if you have freshwater that can support a lot of phototrophs, then they're going to produce a lot of organic carbon. That's going to mean that now the water can support a lot of chemoorganotrophs as well. Those chemoorganotrophs in general would be consuming oxygen, and if you have a lot of them, then you're going to end up with the bottom of the body of water becoming anoxic. All the chemoorganotrophs that are kind of at the top and in the middle have used up all the oxygen, and there's none left to reach the bottom. Then you're going to get a lot of anaerobic bacteria living at the bottom. If you don't have as many phototrophs, then you're going to have not as many chemoorganotrophs, and oxygen will end up being able to penetrate further towards the bottom, so you might not have anaerobes down there, unless of course this is kind of a deep body of water, in which case the bottom would always be anoxic and have anaerobes at the bottom. Then finally, if you do end up with like quite a lot of microbes in the freshwater, that's not necessarily a good thing for the other things trying to live in the water, specifically animals. So if you have a lot of microbes in there, also if you have a lot of aquatic plants in there, they're going to be consuming oxygen, and then there's none left for fish. So you need to kind of have a balance of like not microbial populations in moderation if you're hoping to support fish in that water. Then here, these images, or this image and this graph, are kind of showing like what can happen if you end up having like a lot of organic pollutants in a body of water. Basically what's going to happen is that your microbes are going to explode in their population. Also aquatic plants will explode in their population, and then you get this water that's kind of scummy. It's not nice for fish to live there, because there's not enough oxygen. All these microbes in the plants, they are consuming all the oxygen, leaving none for fish. So perversely, if you add nutrients to water in organic pollution, like in fertilizer runoff or agricultural, or I guess livestock runoff, you can end up making the water not a nice place to live for fish. And then this graph is kind of showing that in more detail. So you've got a lot of lines on here, but here the dotted black line, this would be the actual, this would be the level of nutrients, I guess, specifically ammonium on phosphate, which are usually kind of limiting nutrients in water, in fresh water. And right here, where the line goes up, you had an input of nutrients. So maybe some fertilizer ran off into the water, maybe sewage entered the water, maybe you had runoff from livestock, from a livestock operation entering the water. Those types of runoff will have a lot of organic materials, and generally with them a lot of ammonia and phosphate. So now those levels are increasing in the water. At the same time, you're going to have some nitrifying bacteria taking that extra ammonia and converting it to nitrate. So now you're going to have nitrate levels rising as well. All of these nutrients, the ammonia, the phosphate, the nitrate, these are usable nutrients for microbes. So you're going to see the bacteria levels in that water go up. They're going to shoot up as the bacteria are using all these extra nutrients. You're going to have, as you get more bacteria in the water, you're going to have more BOD, which is biological oxygen demand. Basically, more bacteria, more aerobic chemoorganotrophs means more demand for oxygen. They need more oxygen to support those high population levels. So then oxygen is going to drop. Oxygen is going to drop. Then, after kind of a bit of a delay, you're going to have algae and cyanobacteria, your kind of phototrophs in the water, rising in their population as well. So they're also going to be kind of harvesting all these extra nutrients, and they can reach a pretty high population level. Then they're going to be also producing their own organic carbon, which supports more chemoorganotrophs, which means that they're going to be using more oxygen. But as you proceed down from this input, like if you go downstream from there, then things are going to be kind of leveling off and going back, more closer to normal. So as you go further downstream, the level of extra nutrients, it's going to go back to regular. Then, at some point after that, you're going to have all these extra bacteria, dying off or kind of just not making it that far down in the water. Same thing for the algae. Eventually, their levels will go back to normal as well. Once all these chemoorganotrophs are back to normal, then your oxygen levels are going to be rising back to normal also. But in this kind of area where you had just kind of injected those nutrients into the water, you're going to have a lot of bacteria, not a lot of oxygen, which means probably not a lot of fish. The fish better hope they swim through there fast. Next we move to the ocean, specifically the open ocean. So the open ocean is the part of the ocean that is not close to a shoreline and is not that deep. So this is not like the bottom of the ocean or the depths of the ocean. It's down to about 1,000 meters deep. So, pretty deep, but not that deep in terms of the scale of how deep the ocean really is. The shore of the ocean waters near the shorelines or the coastal regions of the ocean, that's also a common environment. We're not going to talk about it specifically, but it's kind of similar to the open ocean. The main difference would be that you have a lot more nutrients near the shore, and that means that you can get a lot more, a lot higher populations of microbes supported by all those extra nutrients. When you're actually away from the shore in the proper open ocean, you don't have a lot of nutrients present, specifically low levels of nitrogen, phosphorus, and iron. You do have a lot of oxygen and a lot of light, especially at the actual surface. And of course, since it's the ocean, you have a lot of salts. The temperature tends to be not very hot. It does vary depending on where you are in the world, but in general, the temperature of the open ocean would be below the temperature of fresh water. And it also doesn't vary by season as much. The ocean is just such a large body of water that its temperature just isn't going to change, depending on if it's summer or if it's winter. If you're looking at the actual surface of the ocean, there the temperature can change some by season, so there you're going to see the populations of microbes changing some from season to season. But if you're down below the surface, then you're not going to see those temperature changes or seasonal variations in microbe populations. Overall, you have a lot less microbes in the open ocean than what you would have, say, by the shore or in fresh water, partly because it's colder. Well, specifically it's colder than fresh water, and also because there's not as many nutrients as the shore and what fresh water have. But you do have a good amount of microbes still, especially considering that the ocean is so big. There's going to be a lot of prokaryotes, also eukaryotes, protists, and algae. Overall, you have a good amount of oxygenic phototrophs in the open ocean, and they're going to be supporting the rest of the microbes in the ocean by producing organic carbon for them. Overall, there's a lot more prokaryotes in the open ocean than there are eukaryotes. So in general, you have high oxygen levels in the open ocean, which is good because, of course, fish need oxygen to live. But there are some exceptions to that, which would be dead zones and oxygen minimum zones, or OMZs. Dead zones are kind of man-made areas where no fish can live in the ocean. Also, no other animals as well. These dead zones will develop anywhere that you have agricultural runoff into the ocean. We have a large one of the largest in the world actually, in the Gulf of Mexico. It forms where the Mississippi River empties into the ocean. You have a lot of agriculture along the Mississippi River, so there ends up being a lot of fertilizer runoff into it that all dumps into the ocean. So where you have that fertilizer entering the ocean, you have, of course, a lot more nutrients there. So you're going to have population explosions for the microbes in the area. A lot more of those oxygenic phototrophs, like the algae and cyanobacteria, but also a lot more chemoorganotrophs. And it's specifically the chemoorganotrophs that are like the biggest problem, although the algae can get to be a problem as well. Those chemoorganotrophs are consuming oxygen, and if their population levels reach like a really high level, then they consume so much oxygen that they consume all of it, and you literally get an anoxic zone in the middle of the ocean, which can be like huge, freaking huge, and no fish can live there because there's no oxygen there, and we call it a dead zone since it's devoid of life except for microbial life. So these images are showing the dead zone in the Gulf of Mexico. This is, the dead zone visible from space. So you can actually see this is a algal bloom that's formed in the dead zone. So you get nutrients coming in from the Mississippi River that causes this bloom of a lot of algae. These algae are doing, oxygenic photosynthesis, so they are making oxygen, but their presence is going to support a huge number of chemoorganotrophs in the water beneath them, who are going to consume all of the oxygen that was normally present in the water and all of the oxygen that was produced by these algae, creating the dead zone. And here is kind of a graph version of the dead zone. The ocean here is color-coded based on oxygen levels. So, blue is good, and the more intense the color is, the less oxygen you have present there. So these areas will not support fish, which isn't really a good thing for us since, we like to eat fish. In addition to the dead zones, you have oxygen minimum zones. Oxygen minimum zones, or OMZs, are way smaller than dead zones. They're quite small, or at least they should be. And they're also natural, so these are not the result of us polluting the ocean. It's natural for them to be there. Any place where you have nutrients at a higher level than average in the open ocean, you can get an oxygen minimum zone developing, where the extra nutrients are supporting extra chemoorganotrophs that are consuming oxygen. Specifically in these oxygen minimum zones, it's common to have denitrifiers living there. So they would be reducing nitrate and producing molecular nitrogen, nitrous oxide, and nitric oxide. The nitrous oxide and the nitric oxide are actually, bad. Nitrous oxide is a greenhouse gas, and nitric oxide causes acid rain. So it's not positive to have those denitrifiers living there. Also, since the oxygen is, not very high in the oxygen minimum zones, it's hard for fish to live there as well. Normally, these aren't like a huge issue because they're pretty small, but in recent years, we've actually measured that they're getting a good bit bigger, which is worrying. It's good when the ocean has oxygen in it and fish can live there. And we actually think that the reason why these oxygen minimum zones are getting bigger is because of climate change. So as the planet warms, the ocean is warming as well. When the surface of the ocean is warmer, of course the ocean is going to be warming from the surface. The bottom will not be warming. It's the same, just as cold as it's ever been. But the surface of the ocean can warm in response to global warming. And that means that you're going to get stratification in the ocean, just like what you would have in a lake. When you have that stratification, you don't have nutrients mixing between different, I guess, depths of the ocean, and you don't have oxygen mixing either, right? If the surface water isn't mixing with the water beneath it, then you're not going to have oxygen penetrating, down deeper into the ocean. And because you don't have oxygen penetrating, then you're more likely to get an oxygen minimum zone. These denitrifiers actually can't tolerate oxygen. They're anaerobes, so they can only live in, like an oxygen minimum zone or, potentially a dead zone, but they mostly live in the oxygen minimum zones. That brings us to the deep sea, which would be the ocean beneath the open ocean. So if you go further down than 1,000 meters, or like below 1,000 meters deep, that's where the deep sea begins, and it would continue, basically down to the ocean floor. This is definitely an extreme environment. No light will penetrate that deep, so you can't have any phototrophs living there in general. You also have very little nutrients, even less nutrients than what you have in the open ocean. The temperature is cold, the pressure is high, since you're kind of, lower in the ocean and you have a lot of water on top of you. Of course, the further down you go, the higher the pressure gets. And of course, since it's the ocean, salt is high as well. Since there's no phototrophs in the deep sea, basically, the ecosystem or the microbial communities are going to be supported by chemolithotrophs. So you do have chemoorganotrophs there. They're living off of organic carbon that has been produced for them by autotrophic chemolithotrophs. Everybody who lives there grows slowly because the temperature is low and the nutrients are low, and they're basically all extremophiles. Actually, of course, any microbe that is going to live in the ocean needs to at least be halotolerant because it's so salty. So you would have, halophiles and halotolerance in the ocean. But in the deep sea, not only do they need to be able to tolerate the high salt, they need to tolerate the low temperature and the high pressure as well. So you have a lot of psychrophiles and piezophiles. A piezophile is a microbe that prefers a very high pressure. So it's really hard to culture those guys because if you go down to the lower depths of the ocean and get a sample of them, by the time you get to the surface, they're dead. They have to have a high pressure or they die. So the only way that you could do that would be if you had them in some pressurized container that was going to maintain the pressure all the way up to the top, and then of course, as you're transferring them around in the lab, they would need to be just exclusively exposed to high pressures, so it's really difficult to make that happen. It's really hard to culture these guys. Very little is known about the microbes living in the deep sea. It's not easy to go there, and it's not easy to bring what you find back. This graph here is showing the designations of piezophiles. So in green, you have the typical piezophiles that like a high pressure. 1 atm is the pressure that we live at. So on this lower scale here, you can see it's getting really high pressure at the depths of the ocean. The lower you go, the higher the pressure is. But a lot of these guys are living, at several hundred times the pressure that we're under at sea level. Then in addition to piezophiles, you also have extreme piezophiles that like an even higher pressure. Those would be living at the absolute bottom of the ocean. And you have piezotolerance. So some microbes down there actually don't love it to be so high pressure, but they can still tolerate it. And these are the guys that are more likely to be cultured successfully in the lab, since they can still survive and be happy at, our pressure. Beneath the deep sea, you have the deep sea sediments, which is just, the ground underneath the ocean. So there is no oxygen here. Out of all the environments that we've gone through, this is kind of kind of the craziest. There's no oxygen at all. The pressure is very high. You have the whole weight of the ocean on top of you. And then depending on how far deep you are in the sediment, you have the weight of the ground as well. There's extremely little nutrients, even less than in the deep sea. So out of all the environments we've talked about, this environment has the least nutrients of them all. So the closer you are to the surface of the sediment, the more nutrients there are. But it's never going to be more than just a little. So it starts out at a little bit of nutrients in the surface of the sediment, and then the deeper you go, the less nutrients there are. So as you can imagine, there's not exactly a lot of microbes living here. Population levels are very low. Growth probably very slow. As you start going deeper and deeper, at some point you're going to get to higher temperatures. So in general, the temperature is low here, but if you are very deep, the temperature might be higher. We don't know of any eukaryotes that live here, only prokaryotes. They would be, anaerobes and chemolithotrophs. The kind of ecosystem here would be supported by the chemolithotrophs, producing organic carbon for anaerobes. Of course, actually, everybody who lives here would be an anaerobe of some type, since there is no oxygen. We know very little about the deep sea sediments. Out of all the environments we've talked about, we know the least about this one. Even harder to study than the deep sea. The figure here is kind of a summary of all the environments in the ocean. So at the top you have the open ocean, then towards the bottom you have the deep sea, and then of course the ground would be the deep sea sediments. So you got this like green kind of cloud here at the top of the water. That would be where your phototrophs are living. There's a lot of light there. So they're producing organic carbon. That organic carbon is then going to fall deeper into the water. It's supplying nutrients to the other microbes in the open ocean. Some places you may have an oxygen minimum zone where the oxygen ends up being a little lower. Well, I should say the nutrients end up being a little higher. Then you get chemoorganotrophs consuming those nutrients and consuming the oxygen that's present, creating a little area where the oxygen is depleted, and then you're going to have denitrifiers living there, producing nitrous oxide, which then rises, as well as nitric oxide, which both rise to pollute the atmosphere. Then in the deep sea, you have mostly chemolithotrophs supporting the ecosystem. You're going to have in some places hydrothermal vents that are kind of spewing minerals from deep in the earth. A lot of those minerals are useful for chemolithotrophs. They can use them as electron donors. So that's kind of a primary source of material for the chemolithotrophs in the deep sea. In the deep sea sediments, you also have minerals which are going to be used by chemolithotrophs in anaerobic respiration. And in some places you might have like a coal bed, which would have more nutrients associated with it, so probably more microbes living there, or a little pocket of hydrated gas, for instance, could be methane. So you might have little bits of methane seeping up from the ground, and of course some microbes that are happy to live off of that methane, specifically the methanotrophs. And the final environment that we'll talk about is the hydrothermal vent, also known as a deep sea vent. So these are found scattered across the ocean floor. In some places they're caused by geothermal activity, sort of like a hot spring or a volcano. So you have kind of stuff like hot minerals coming up from the depths of the earth, coming up through these vents. In some places it's actually not like that at all. You just have some minerals from the deep sea sediments that have become exposed to the water, and they're actually having a reaction with the water that creates heat. So it would be an exothermic reaction, sort of like the way that like a hand warmer would work, but more intense. At all these hydrothermal vents, you're going to have, of course, a very high pressure because you're at the bottom of the ocean. And since you're in the ocean, you're going to have high salts, and you're going to have no oxygen. The pH of the vents will actually vary depending on what type of vent it is, and the temperature will vary as well. Of course, it's always going to be, hot. Some of them are actually not super hot. Some of them are just kind of warm, and some of them are super hot. They can even get to above boiling. So normally you cannot get water to be, higher in temperature than the boiling point. But since you're at the bottom of the ocean here and the pressure is very high, it is actually possible to get the water at the bottom of the ocean, up above the boiling point. So some of these vents have extraordinarily hot water, far above the boiling point. Of course, you're not going to have microbes living in the very, very, very hottest water that is possible to have coming out of these hydrothermal vents. But you're going to have, kind of the cold water around the vent mixing with the really hot water coming out of the vent, and that creates, a more moderately hot water that the microbes can live in. If the vent is really hot and is caused by geothermal activity, we call that a black smoker because it's emitting all these minerals that form something like a black smoke. It's just not properly a smoke because this is all happening underwater, but clouds, plumes of black crap. That type of vent would have a low pH, so it would be acidic. If it's a vent that's being created by a reaction between exposed minerals and the seawater, that type of vent is going to be high in pH in general. So it would be like basic or alkaline. Also, those vents, the exposed mineral vents, will tend to last a really long time. They're very stable. The geothermal activity vents, they can last a long time, but just not as long. In general, you would have chemolithotrophs living around these vents, and they would all be prokaryotes. And of course, most of them would be thermophiles or hyperthermophiles. The specific type of chemolithotroph that you have there is going to depend on specifically what type of mineral is coming out of the vent. And a lot of these guys, even though they're thermophiles, they actually can tolerate cold water that's further away from the vent. They won't grow in it. They can't reproduce there. It has to be hot for them to reproduce. But they can at least, not die. So if the currents if the ocean currents carry them away from their vent, they can just kind of like subsist in a dormant state. And they might end up at a different vent, and then when it's warmer, they'll kind of, blossom again and grow. And the diagram is just showing kind of a summary of the hydrothermal vents. On the right, you have a black smoker with different minerals coming up from the depths of the earth and coming out through the black smoker vent. Some of these minerals will end up falling to the seafloor and forming the sediments, and then that supports chemolithotrophs living in those sediments. Then here you have a warm vent, so it's not crazy hot. This one is actually going to be caused by a reaction between exposed minerals and the seawater. So yeah, you would have different microbial populations at each of these different vents because they have different minerals associated with them.